Hey, how's it going, guys? Mr. Binder here. Um, this section of material that we're going to look at is Chapter 3 in our textbooks, or better yet, an introduction to metabolism. Now, metabolism is the sum of all the reactions that take place in our cells. And this is important, uh, again, because we need to lay the foundation for some groundwork. Uh, Knowing this information, we're going to have a better understanding of cellular respiration and photosynthesis. Obviously, we're going to be taking a look at these two topics later on. These are both cellular processes. Both require energy. And essentially what metabolism is the sum of all the reactions that take place in our cells. Well, chemical reactions require energy. And energy is obviously the ability to do work. Notice the examples of different types of energy that uh, I've got here for you guys. Chemical energy, electrical, mechanical, light, and thermal. These are all examples of different types of forms of energy, and we will take a look at these later on. All right, taking a look here, metabolic rates. It's the amount of energy an organism uses over a specific time. And obviously, when we do any type of exercise, our metabolic rate will increase. Taking a look at the basal metabolic rate, individuals that are resting will be able to find their basal metabolic rate. It is the actual amount of energy, the least amount of energy required to perform chemical reactions in a cell. And taking a look here, the higher percentage of body fat reduces your metabolic rate. Energy can exist in two states, kinetic and potential. We're going to use these two terms closely as we take a look at this topic. All right, kinetic energy is just motion. When molecules, ions are moving in the blood, or any type of energy that's moving. Okay? Potential energy is stored chemical energy. Stored chemical energy, especially in this chapter, we're going to take a look at energy that's stored in bonds, chemical bonds. Energy obeys laws. We're going to use two laws here to describe energy, and they're known as the first and second law of thermodynamics. Now, the total amount of energy in any closed system is constant. This means that this system here is isolated from its environment. There's no incoming energy or outgoing energy. It's always it's isolated from from an environment. Energy cannot be created or destroyed. Okay? This is why you guys were balancing those chemical reactions in grade 10. Okay, the reactants need to be the same as the products. All right, but energy can be converted from one form to another, and we will see that, especially in cellular respiration and photosynthesis. And again, we see that if a system gains an amount of energy, another system has to experience a loss of energy. This makes sense because energy cannot be created or destroyed. Energy obeys laws. Again, photosynthesis, respiration. This is important. These laws of thermodynamics are important when describing photosynthesis and cellular respiration. Okay? Photosynthesis obviously uses sunlight. Energy here is the sun. And plants, this is the system that occurs in plants, converts sunlight to chemical energy. And that chemical energy will be stored in chemical bonds. Now, cellular respiration will convert chemical energy, and we will use that and convert it into mechanical energy, for example, a muscle contraction. The conversion of energy, it depends on breaking and forming of chemical bonds in a reaction. Now, energy is absorbed to break bonds, and energy is released when new bonds form. Now, these two statements here are very important in terms of figuring out whether there was a release of what will be known as free energy. This is important when we are trying to describe chemical reactions. Bond energy. Bond energy is the measure of strength or stability of a covalent bond. All right, there's energy in between bonds here. So these hydrogens are going to be sharing electrons. Remember, a covalent bond is the sharing of electrons. Well, that bond that's forming between those two atoms, there is an amount of energy in that bond. Now, in terms of a chemical reaction, what happens is bonds are broken and bonds are reformed. So that means that there is a conversion of or transfer of energy from reactants to products. Now, different amounts of energy are required to break different types of bonds. Notice here that each bond that you see on the left requires a specific amount of energy used to break that bond. When taking a look at a chemical reaction, the activation energy here, that is the required amount or minimum amount of energy needed to break a bond and start a chemical reaction. So what we could do is take a look at the amount of bond or the different types of bonds in the reactants. We could calculate how much energy is required to break those bonds, and that would be your activation energy. With any chemical reaction, an activation energy is required. Okay? This will start the chemical reaction. Now, once the chemical reaction's started, the reactants will start to change. Their bonds will be broken. Okay? That they go into a transition state, and then those bonds will reform in the products. Now, there's two different types of reactions that we can use to describe a chemical reaction: an exothermic reaction or an endothermic reaction. Essentially, an exothermic reaction is a net release of energy. So the amount of energy that was stored in the reactants here is obviously more than the energy that is now stored in the products. Having said that, there was a release of energy as the new bonds formed in the products. On the flip side, the products now have more energy stored in the bonds than the reactants, meaning that there was an absorption of energy here. And these are two different types of reactions: endothermic and exothermic. Exothermic reactions are reactions that obviously release energy and they're hot, all right, or they release heat. Endothermic reactions, they absorb heat, so they're the I guess the ice pack when you have to break it up, and next thing you know, it's nice and cool to the touch. But we can calculate in any chemical reaction the amount of energy that's released or absorbed with this reaction here. Bond energy absorbed minus bond energy release will give you your net energy, whether it was released or absorbed. And we can use that to understand if a chemical reaction was exothermic or endothermic. All right, the second law of thermodynamics. Essentially what happens is in every energy transfer conversion, some of the useful energy becomes unusable. This unusable energy increases entropy, okay, of that system. Now essentially entropy is just a degree of randomness within a given system. So we take a look here and we can use entropy in this picture. As entropy, okay, a molecule will have low entropy because it is more ordered. So taking a look here, when we compare this glucose molecule to the water and carbon dioxide molecules, the glucose molecule here is more ordered. Okay? There's high free energy, so that means that because it is more ordered and is less random, it has a low entropy. Now, as that molecule breaks apart, and this is basically, we're starting to describe cellular respiration here. As this molecule breaks apart into carbon dioxide and water, we are going from a larger single molecule to smaller molecules, multiple molecules. And essentially what we're doing is, or what's happening as this chemical reaction proceeds, is that things are becoming more disordered, okay, or more stable. Essentially there's low free energy, but there's high entropy. So we're increasing the randomness, okay, or things are becoming more disordered. Essentially what's happening here is entropy will be increased. And that's exactly what entropy is. Right? Entropy will increase when solids react to form liquids or gases. Liquids react to form gaseous products. Okay? The total number of product molecules is greater than the total number of reactant molecules. So essentially what happens, we have one molecule here broken apart into hundreds of molecules here. When this occurs, an increase in entropy is used to describe this system when it moves from this way to this way. Right? And the reason why we're using this entropy, or we're trying to understand entropy, it is related to cellular respiration and photosynthesis. When a glucose molecule is broken down into water and carbon dioxide, entropy is increased. And on the flip side, when water and carbon dioxide are used to form glucose in photosynthesis, entropy is decreased. Spontaneous versus non-spontaneous change. Essentially what could happen here is we can predict whether a chemical reaction will occur, okay, without the continuous input of additional energy. And what that means is once that activation energy is available, a chemical reaction will proceed. Now there are some chemical reactions that require a continuous supply of energy, like trying to boil a pot of water. You need a constant supply of energy. The example here is iron. Iron reacting with oxygen and water to form rust. That's a spontaneous change. Okay, rust forming back into iron, that is obviously non-spontaneous. Spontaneous change or spontaneous chemical reactions, these are your favorable chemical reactions. They'll continue to occur on its own once the activation energy has been met. Okay? There's no additional energy required after the activation energy. There's obviously three factors that determine whether a reaction will be spontaneous. Okay? If there's a release of energy, then there will be a spontaneous change. An increase in entropy. Increase in entropy means from a big molecule to smaller molecules, and at high temperatures. Non-spontaneous. This is the other way. Essentially what happens here is there needs to be a continuous supply of energy. Okay? Additional energy is required after the activation energy has been met. The amount of activation energy that has been met for the actual chemical reaction. Some factors determining whether a reaction will happen. Okay? Endothermic changes. So essentially, there's an absorption of energy in the bonds. Okay? A decrease in entropy. That means that smaller molecules are getting put together to form a larger single molecule. And this is lower temperatures. So taking a look here, we see that there is an absorption of energy in the products here. Okay? An absorption of energy. And again, this is a not favorable chemical reaction. I mean, they'll still happen. The example obviously would be photosynthesis. Plants use the sun all the time, right, when it's available, to perform this specific type of reaction, a non-spontaneous change. Gibbs-free energy. This energy that's not lost during a reaction. Okay? Essentially, Gibbs-free energy, we are going to use Gibbs-free energy to describe spontaneous and non-spontaneous changes and chemical reactions. Essentially, Gibbs-free energy is important during cellular respiration because this is the energy that will be used, okay, in the form of ATP. All right. So when our cells break down a glucose molecule, there's a lot of energy in those bonds. When glucose is broken down, that energy from the glucose will be stored in the form of ATP. And this ATP obviously is the energy currency of the cell and will be used for all cellular processes and basically just help the cell to perform its function in terms of driving chemical reactions forward. Okay. So taking a look here, gives free energy is responsible for synthesis of molecules, a reproduction, movement, essentially all functions of the cell. It's represented by G absorbed minus G released. Okay, and we can figure this out in terms of an actual calculation. So taking a look here, okay, the example is we have A plus B gives you our products. Okay, these are reactants here. Okay, reactants are A plus B, products are C plus D. Calculating the bond energies within the reactants A and B, we can calculate how much energy is absorbed in the bonds of the reactants. And the example here is 300 kilocalories. Again, we can calculate the bond energy that has then been released, okay, as new bonds have formed. And in products C and D here, it's 450. Now taking a look there, we can calculate this if we take the absorbed minus the released. Here they have it released minus absorbed, which is fine. Okay, I want you guys to use the equation that I have up there, only because the math will obviously it will help you to describe, okay, a chemical reaction. All right, when you do absorb minus released, again you'll get a negative number. Okay, here the way that this example occurs, 450 minus 300 doesn't give you that negative value there. Okay, that negative is very important in terms of describing whether this chemical reaction is an exothermic, endothermic, exergonic, or endergonic reaction. Okay, essentially bond energy in the products is larger than the bond energy in the reactants. If this occurs, delta G has to be negative. Delta G meaning the free energy of the chemical reaction. That means that this chemical reaction is exergonic. There's a release of energy. That's why we get this negative value. If the bond energy, okay, if the bond energy in the products was less than the bond energy of the reactants, okay, delta G would be positive. Okay, and this would describe a endergonic reaction. All right, and if we take a look here, bond energy of the products, let me see. Oh, that's a big, equals the bond energy, okay, of the reactants. Then delta G is 0, okay, because the chemical reaction did not occur. Again, you're going to have to use this equation here, okay, delta G absorbed minus delta G released, to calculate whether or not a chemical reaction was endergonic or exergonic. Please understand that this example put the products first rather than last. So I want you to put the absorbed first and then minus released. Spontaneous. So again, this is the negative values. If we see that there is a negative G as we calculate the energies in the products and the reactants, that would obviously be a spontaneous change or an exergonic reaction, a release of free energy. Okay. The example that I have here is the breakdown of glucose, or the oxidation of glucose. Okay. There is a lot of energy released as a glucose molecule changes to carbon dioxide and water. Again, glucose reacts with oxygen. Okay. The reason why I give you this example is this is the reaction for cellular respiration. All right, glucose, lots of energy in glucose. Again, there is some activation energy that's required here to break these bonds or to start the chemical reaction. But once that chemical reaction has started, glucose will then transform. Okay, there will be a transfer of energy or a release of energy as glucose changes from a reactant to a product here, carbon dioxide and water. Right? Now, the energy here, this release of energy, okay, this will be stored as ATP, and this is essentially what occurs during cellular respiration. Non-spontaneous, okay, this is the flip side. This is now we're describing photosynthesis. This is when delta G is now positive. Essentially, the free energy, okay, of products is more than the free energy of the reactants, and we can use this. We can describe this by simply calculating the bond energies in the reactants and the bond energies in the products, and then comparing the two. Right? So photosynthesis obviously needs activation energy, much like cellular respiration would. Again, it needs a continuous supply of energy in order for this reaction to proceed, because it requires energy to form the bonds here in glucose. Okay, so this is a non-spontaneous change or non-spontaneous reaction. Always needs energy. And where's that energy coming from? Well, when the sun comes out, the plants will continue to use the sun's energy to convert carbon dioxide and water in the form of glucose. Again, the reaction here is essentially the exact opposite of cellular respiration. So we would see that delta G is positive, the same exact number, 2,870 kilojoules per mole of free energy that's stored in the bonds. Our cells use both types, okay, exergonic and endergonic reactions. Okay, our cells will use ATP to drive endergonic reactions. All right, and our cells carry out, again, thousands of chemical reactions. Okay, when our cells need energy, it's going to use ATP. All right. Keep that in mind as we try to describe how our cells can drive endergonic reactions. All right. Again, endergonic reactions require a continuous supply of energy, and this energy will come from ATP, or the hydrolysis of ATP. All right. Now, coupled reactions. Essentially, what coupled reactions are is the release of energy from an exergonic reaction can drive endergonic reactions. Okay? So what does that mean? Well, the hydrolysis of ATP will be used. So when ATP is then essentially used in a chemical reaction, the energy found in the bonds of the ATP molecule will be used to drive chemical reactions that require a continuous supply of energy. Right? So combining more than one chemical reaction here, all right, an exergonic and an endergonic, we call this coupled reactions, only because when both of them are coupled together, the amount of energy that's going to be used in the exergonic reaction is enough to drive the endergonic reaction here. Okay, so we have A gives you B plus C. All right, this is our first chemical reaction. This is a spontaneous change because the negative value there, delta G, so there's a release of free energy throughout this reaction. Again, D plus E gives you F. It requires 4 kilojoules per mole of energy, okay, in order for this reaction to occur. Well, chemical reaction number 1, all right, there's a release of energy. Now let's do the math. What's negative 5 plus 4? Okay. Again, when we couple these reactions together, all right, the net energy here is negative 1. There's still a release of free energy. So essentially what's going to happen is the release of the free energy from the reaction 1, okay, will be used to drive the reaction in the chemical reaction number 2 here. All right. And this is known as a coupled reaction. And essentially what our cells do is it uses ATP. The free energy found in ATP will be used to drive endergonic reactions. They require continuous supply of energy. Catabolic versus anabolic. These are, again, terms that we can use to describe a chemical reaction. We've heard exothermic, okay? We've heard endergonic, exergonic. All right, now we're using these terms catabolic versus anabolic. What does that mean? Well, catabolic reactions are essentially very similar to spontaneous reactions. Okay, this is a breakdown of big molecules into smaller ones. So again, spontaneous change, we see an increase in entropy. Okay, there's a release of free energy. Okay, so again, delta G will be negative. Flip side, anabolic reactions. All right, we're taking smaller molecules and we're using those smaller molecules to build a bigger molecule. All right, this is essentially a non-spontaneous change. Again, free energy is required continuously in order for this process to occur. Okay, and delta G will be positive here. Okay. ATP, again, very important, because the amount of energy that's going to be used to drive specific chemical reactions, ATP can essentially use, well, the amount of energy found in ATP is enough to drive chemical reactions in a cell. And what does that mean? Well, taking a look at the picture here, the sodium-potassium pump. This is an example of active transport. So pushing molecules across the membrane against the concentration gradient. How can it do that? It requires energy. Well, that energy comes from ATP. So this here is a coupled reaction. Okay? A coupled reaction seen between this transporter protein and ATP. There is a transfer of energy from ATP to the pump. That transfer of energy will drive that chemical reaction that we see occurring here. Okay? When sodium is pumped across the membrane, okay, against the gradient. All right. Now there are other forms of energy, GTP or UTP, but what I want you guys to focus on is ATP as being the energy currency of the cell. All right, where does the energy from ATP come from? Well, it comes from a glucose molecule. Our cells break down a glucose molecule, and using that energy from that glucose, we will store it in the form of ATP. All right, there's a lot of high energy here in the phosphate groups. Oh, a lot of high energy. And when that phosphate group breaks off, this is known as the hydrolysis of ATP. All right, we end up going from ATP to ADP plus phosphate, essentially, and a release of a lot of energy. About negative 30.5 kilojoules per mole. All right, that energy there will be used once the hydrolysis of ATP occurs. Okay, in the reactions here in the picture, we have ATP. All right, the hydrolysis of ATP, meaning that a water molecule is used to break up ATP. That hydrolysis of ATP, again, breaking apart that phosphate group. All right, and we end up with ADP, inorganic phosphate, and a lot of energy. That energy, again, is negative 30.5 kilojoules per mole. That energy will be used to perform or used in endergonic reactions, reactions that require a specific amount of energy. Again, we see here ATP will be used in terms of coupling reactions. Okay, so we have the picture here. Glutamic acid and ammonia, all right, forming glutamine. Glutamine is an amino acid. Okay, so without ATP, it requires obviously a continuous supply of energy, all right, plus 14.2 kilojoules per mole. Well, when we add ATP into the mix, we see that ATP, or the release of ATP, the amount of energy here in ATP, negative 30.5, okay, is obviously enough to drive this chemical reaction here. So we're coupling the hydrolysis of ATP, okay, to reaction 1 here, all right, glutamic acid and ammonia, all right. We're coupling the hydrolysis of ATP with this reaction, in order to get glutamine. Okay? And we can see that this process will occur once ATP is broken down and the release of free energy is provided for that reaction to occur. And again, we can do that simply with the math here. 14.2 obviously plus negative 30.5. The net delta G after these two reactions occur is negative, meaning that this reaction or both reactions will occur spontaneously. Okay? Regeneration of ATP. Essentially, this is cellular respiration. Our cells need to break down glucose in order to continuously make ATP. We need ATP to drive these chemical reactions in our cells. All right. So how do we obtain the energy to create ATP? Again, food, breakdown of carbs. All right. Carbs is basically your glucose molecule. All right. Our cells can also break down fats and proteins, but they're all sources of energy. Okay? We're going to be taking a look at glucose. Glucose is the molecule, again, that's the major playmaker here in cellular respiration. All right, and it's the energy that's found in glucose that our cells will use to store as ATP. Again, ATP cycle here. Okay, again, notice how many times ATP is broken down and reformed. All right, 10 million times per second in our cell. That's a lot of energy that's being used to drive specific chemical reactions just within a cell. We're made up of trillions of cells. So we need to continue to supply our cells with this energy. Okay, and obviously the energy that we get this from, where we get this from would be in our glucose, okay, or from our diet. We break down food, okay, we absorb it in our body, we essentially transport it into the cell. The cell will break down that food or glucose and store that energy in the form of ATP. Enzymes are very, very important here, okay, because we learned about enzymes earlier. Enzymes lower activation energy, so the amount of energy that's required to drive a specific chemical reaction forward. Okay, so essentially what enzymes do, enzymes are very, very important in our cells because enzymes will lower the amount of energy that's required for a chemical reaction to proceed. Again, the name of an enzyme ends in ACE. So whenever you see ACE at the end of anything, it usually means that this is an enzyme. The example here is maltase. Maltase is the enzyme used to break down maltose into its parts, glucose and glucose. So we're seeing the breakdown of a disaccharide here into its two monosaccharide components. Okay. Again, what provides activation energy for a chemical reaction? Well, thermal energy. Okay, so combustion reactions here. Well, essentially the example is a spark used to create a flame here on this propane torch. All right, but this isn't good for a cell. Essentially, increases in temperature is bad in biology, and we've seen this because if there's an increase in temperature, it throws off our own homeostasis. Essentially, the enzymes won't function properly, and we'll die. So again, the combustion of glucose is something that our cells need to control. Okay, so how do our cells do that? Well, it's broken down, and we'll talk about it. Broken down into a series of steps. All right. But we're still talking about enzymes here, so I can't get ahead of myself. But enzymes lower activation energy. Okay, enzymes lower activation energy. How do enzymes reduce activation energy? There's three different ways. Essentially, bringing molecules together. All right, so substrates and the active site, bringing the substrate together in the active site of the enzyme here. All right. Essentially, an enzyme can expose molecules to altered charges of environments. So again, could change the polarity of the molecule. Okay. And again, enzymes can also change the substrate or the shape of the substrate. Essentially, it can bend the bond that's found between, these atoms of this reactant here, and when it bends that bond, it can weaken it. Weakening that bond obviously would not require the same amount of energy used to break that bond. And food as fuel. All right, we see here glucose. Glucose is a great molecule because there's a lot of carbon-hydrogen bonds. All right, carbon-hydrogen bonds. There's a lot of energy in between these bonds here. And again, we can see that when we do break down a glucose molecule, there's a release of a lot of energy. Okay, and our cells need to capture that energy and store it in the form of ATP. All right, so essentially what we're taking a look at here is the carbon-hydrogen bond. There's a lot of energy in between the carbon-carbon bonds and the carbon-hydrogen bond, and this is a really good bond for our cells in terms of breaking it down and capturing that energy. Again, I was getting ahead of myself, but essentially, the combustion or oxidation of glucose in our cell needs to be controlled. Okay, controlled. And how is this controlled? How is the breakdown of sugar or glucose controlled in our cell? Essentially, it's controlled in our cell by using a series of enzyme catalyzed reactions. That's why there's about 8 or 10 reactions in glycolysis alone. Again, with the Krebs cycle, another 8 enzyme catalyzed reactions that are used. Okay, it's a series of steps to control the amount of oxidation. We want to control the oxidation of glucose because we want some stability in the cell. Okay, we want some stability. We don't want that release of free energy immediately. If there was, that would be bad for the cell. Right? So oxidation of glucose in our cells is an exergonic reaction, but it is a controlled oxidation reaction, meaning that glucose will, or our cells will capture the energy from a glucose in a series of steps. And those series of steps is cellular respiration, is glycolysis, is pyruvate oxidation, is the Krebs cycle. Okay, those are all steps that are necessary in terms of capturing the energy in a glucose molecule. The last slide here, this is important because when our cells do start to break down glucose, it needs energy carriers. So the energy from glucose, needs to be kind of harnessed and transported. Well, where is that energy going to be kind of temporarily stored? Well, with the help of these energy carriers. Okay, and this is where we're going to be introduced to NAD and FAD. All right, and these are your molecules. These are your co-substrates. Okay, essentially what these molecules will be used for is to take the electrons from a glucose and transport them over to the electron transport chain. This is a transfer of energy. These are a series of redox reactions. Okay, and the transfer of electrons from glucose to NAD. All right, and then finally the electron transport chain will allow for the not only the controlled oxidation of glucose, but the transfer of energy, and we will see this transfer of energy stored in the form of ATP when we do discuss it. All right, so please remember NAD and FAD because they are two important molecules. They're the two key players here when our cells do start breaking down glucose and form ATP.